When cells get too big to function efficiently, they must divide. Like the cartoon illustrates, when they divide, they produce two genetically identical copies. Did you know that twins are genetically identical? The division of the nucleus is also called mitosis. It only happens in eukaryotic cells because they're the only cells with a nucleus. In prokaryotic cells like bacteria, the process is simpler and is called binary fission. Cells divide during the M phase of mitosis. Mitosis is the division of the nucleus and cytokinesis is the division of the cytoplasm. But what do you need new cells for? Aren't the ones you have good enough? Well, not always. Cells get worn out just in the course of performing their functions, so existing cells must divide in order to produce new cells. More cells are needed if you are to grow larger as well. Since we've already established that it's inefficient for cells to get very large because of the demands on their DNA and difficulty transporting materials, when we grow, we have to produce new cells. Growing just isn't for kids, though. If you injure yourself, like getting a scrape on your knee or breaking your bone, new cells replace the damaged cells. Single-celled organisms also undergo mitosis. This is how they reproduce. Single-celled organisms without a nucleus undergo a process called binary fission to make offspring. Mitosis is usually divided into 4 phases: prophase, metaphase, anaphase, and telophase. The first and longest stage is prophase. Some textbooks break it into prophase and prometaphase because it's so long. During most of a cell's life, its DNA is associated with proteins called histones, which coil up the DNA inside the nucleus. There's 3 meters of DNA inside of your cells, and your cells are so small you can't even see them. So the proteins that help DNA physically fit inside of the nucleus. While a cell is not dividing, this protein DNA substance is called chromatin. It's sort of like a ball of yarn a cat is played with. Some parts might be tightly coiled if the cat hasn't pulled that apart yet, and other parts might be fairly loose. However, during cell division, chromatin condenses, forming chromosomes. Chromosomes look like X-shaped structures and help organize the DNA. Chromosomes contain 2 copies of DNA. One copy was made during the S phase of the cell cycle. Also during prophase, the spindle begins to form and chromosomes attach themselves to the spindle fibers. The green lines in this picture are the spindle. Each individual green line is a spindle fiber. You can see how the spindle attaches to each side of the centromere, or center of the chromosome, so that it can be pulled into 2 identical halves. The way they're able to do this is by shortening the microtubules that make up the spindle fibers. 2 other important events happen during prophase. The nuclear membrane breaks down, and centrioles, which are organelles that coordinate cell division, move towards opposite sides of the nucleus. The centrioles were formed during the G2 phase of mitosis, and they move to opposite sides of the nucleus during prophase. See, I told you prophase was a busy time. Here are a few pictures of cells that are all in prophase. The cell cycle is a continuous process, so it might seem like some cells are hard to put into distinct categories. The purple cell on the left and the topmost labeled cell on the right are both very early in prophase. The nuclear membrane is still mostly intact, and you're just starting to see the formation of chromosomes. In interphase, the nucleus of the cell, which are the dark centers in these pictures, look like a solid circular blob. It may have a dark, dense dot in the center as well. This is the nucleolus. During prophase, spots of white show up because the chromatin is condensing to form chromosomes. Long, stringy-looking chromosomes are more visible towards the end of prophase. The second phase is fairly straightforward and easy to pick out. The chromosomes are lined up in the center of the cell. One sister chromatid faces one direction, and the second sister chromatid faces the other way. This way, when microtubules shorten, each side of the cell will have one complete set of DNA. The imaginary line through the center of the cell, where the chromosomes line up, is called the metaphase plate. In plants, this is where the new cell wall will eventually form. In anaphase, the chromosomes are pulled apart at their center when the spindle fibers shorten. The center of a chromosome is called the centromere. On each side of the centromere is an identical copy of the DNA, so that each new daughter cell gets all of the DNA. The final stage of mitosis is telophase. I like to think of it as the backwards process of prophase. For example, during prophase, the nuclear membrane disappears, and in telophase, it reforms. In prophase, chromosomes condense to form chromosomes. In prophase, chromatin condenses to form chromosomes, and in telophase, the chromosomes disperse themselves and go back to being chromatin. The spindle, which was formed during prophase, now breaks apart during telophase, and the nucleolus also reforms during telophase. The final stage of cell division isn't technically part of mitosis. It's part of the M phase of the cell cycle, though. Mitosis is the division of the nucleus, which is what I just explained, and cytokinesis is the division of the cytoplasm. Many times telophase and cytokinesis can happen at the same time. Cytokinesis is different in plant and animal cells because of the presence of the cell wall in a plant cell. The formation of the cell plate is in the same location as the metaphase plate and is unique to plant cells. This is what's going to become the cell wall. Instead of forming a cell plate, animal cells pinch their flexible cell membrane inwards to produce a new cell. And there you have it. Once going through prophase, metaphase, anaphase, telophase, and cytokinesis, the cell has completed division, and now there's two new identical daughter cells.